So today we'll discuss about crossing over.
Yes, sir.
Okay. So this crossing over takes place in meiosis. In meiosis, there are two substages.
Meiosis first and meiosis second.
Meiosis 1 and 2. Okay. In meiosis 1, there are again four phases.
Prophase, metaphase, anaphase,
Telophase.
telophase.
Phase 1, metaphase 1, telophase 1.
Metaphase.
Here also same, but we'll write 2. Phase 2.
Yes, sir.
Metaphase 2. Anaphase 2. Telophase 2. Okay. So here in prophase 1, prophase 1, there are again 5 substages. Leptopene, zygotene, pachytene.
So prophase first types.
In prophase 1, again there are substages.
Okay, sir.
In prophase 1, there are again 5 substages. Leptotene, zygotene, pachytene, diplotene,
Dikinesis.
Crossing over takes place in pachytene. Crossing over takes place in pachytene.
Pachytene.
So this phase, pachytene, is a phase where crossing over takes place. Okay.
So crossing over is take place is pachytene.
In pachytene. So crossing over will occur in pachytene.
Yes.
And when we come to the process of crossing over, in the process of crossing over, so first we should understand about Homologous chromosomes.
Homologous.
We should understand about homologous chromosomes. Homo means same. Homo means
Same.
same. I'll just explain you with an example.
Yes, sir.
So total, if you take humans, so there are 23 pairs of chromosomes, or we can write in form of 46. Okay. If you
Yes,
write
sir.
23 or 46, same. 23 pairs or 46,
46 chromosomes.
same. Okay. So here if you take 23 pairs, so you can write like this. First pair, second pair, like that you can write on up to 23rd pair. Okay. You can write like this.
Sir, what is homologous chromosomes, sir? Homologous chromosomes is
That's
same.
what I'm explaining. Okay.
Yes, sir.
So in first pair, so there will be 2 chromosomes. There will be
2 chromosomes.
2 chromosomes. So there will be 2 chromosomes. The first pair, there will be 2 chromosomes. Okay. So these 2 chromosomes, so one chromosome is from one parent. Suppose if this chromosome is from mother,
Yes.
it is called
Mother
maternal
Maternal.
chromosome.
Chromosomes.
Automatically, other one will be from father.
Parental
Yes.
chromosomes.
Paternal chromosome. So paternal means father.
Up. Yes, sir.
Maternal means
Mother.
mother. Okay. So in one pair, one chromosome will come from mother and another chromosome come from
Parent. Father.
so these chromosomes will have alleles. Alleles means suppose if you take height. Okay. Just for example, take height. Suppose if this chromosome is having capital T, this chromosome will have small t. This chromosome will have small t. Okay.
Yes, sir.
So they have same set of genes. Okay.
Yes, sir.
Suppose here they have capital D, here they will have small d.
Small d.
That means So these chromosomes will have alleles of the same gene. Okay.
Yes,
These
sir.
chromosomes are called homologous chromosomes. So these chromosomes are called
Homologous chromosomes.
homologous chromosomes. So each pair of chromosome will have one maternal chromosome and one paternal chromosome. So each pair will have one maternal and one paternal chromosome.
Maternal chromosomes.
So this pair of one maternal chromosome and one paternal chromosome called homologous chromosomes. This is called homologous
Homologous
chromosomes.
chromosomes.
So here this, so the process of crossing over, if you take, so crossing over will take place between these two homologous chromosomes. So crossing over will take place between homologous
Two
chromosomes.
homologous chromosomes.
Okay. Crossing over never occurs between first pair of chromosome and second pair of chromosome. Okay. It occurs only between first pair, between
Yes, sir.
between third pair. Like that it occurs. Okay. So this crossing over, so if you take the process of crossing over, so the process of crossing over has 4 steps. So first step is called
Synapsis.
synapsis. Second step is
Tetrad.
formation. Third step, so the third step is exchange of chromatics. Third step is
Each
exchange of chromatics.
exchange of chromatin.
Fourth step, disjunction. Okay. So the process of crossing over has 4 steps: synapsis,
Tetrad formation.
tetrad formation, exchange,
Exchange of chromatid.
disjunction.
Disjunction.
Okay. So first we'll go one by one. First we'll go one by one, the process of the steps one by one.
Yes, sir.
So if you come to the first step,
Synopsis.
so synapsis means, in synapsis what will happen is, so in synapsis the homologous chromosomes will combine, they will attach with each other.
So synapsis is homologous. Chromosomes are attached.
Chromosomes will attach with each other. So the process of attachment of homologous chromosomes is called synapsis.
Synapsis.
Okay.
Yes,
So here
sir.
2 chromosomes are attached. So this both attached one combinedly, it is called bivalent. So
Sir, what is bivalent?
bivalent is this total is called one bivalent. This total is called bivalent. Bi means how many?
Two.
Two. So here two chromosomes are attached with each other. So this is called bivalent.
Yes, sir.
This is called
Bivalent.
bivalent. Okay. So this bivalent is formed during zygoting. I already told 5 phases are there in prophase 1. Okay. In the 5 phases, synapsis takes place in zygote. Synapsis takes place in
Zygote.
zygote. Okay. And when you come to the after synapsis, so after synapsis, what will happen? Okay. After synapsis, the next stage is tetrad formation. So the next stage is
Tetrad formation.
tetrad formation. Tetra means how many?
3. It's a 4, 4.
Tri means 3. Okay.
Yes, sir.
Tetra means,
Tri is 3.
so tetrad formation means what will happen here is, so in bivalent, so two chromosomes are combined. In synapsis, two chromosomes are combined. Okay. So just for our understanding,
Sir, don't synapsis has 2 chromosomes is attached. Here,
so
here
two
also 2
chromosomes.
chromosomes is
No, no,
attached.
here I will tell the next step what will happen.
Yes.
Okay. So just for our understanding, I will show difference between paternal and maternal chromosome. Okay. So just think that it is maternal chromosome.
Yes.
I'm writing here.
Sir, only maternal chromosomes.
Okay, this is paternal.
Okay, sir.
So this darkened one is
Maternal.
maternal. Okay. So this light one is
Paternal
paternal.
chromosome.
Okay. So the center circle one is called centromere. Am I correct?
Yes, sir.
It is called centromere. And this one is called arm. A chromosome will have one centromere
And 4 arms.
and arms. Okay.
Yes.
So in tetrad formation, the arms of the chromosome will split. Only arms of the chromosome will split. It means they will divide.
Yes, sir.
So when arms of the chromosome split,
It is a split of 8 parts.
yeah. When arms of the chromosome will split, will get chromatids. will get chromatids. These are called
chromosomes.
chromatids. This one is one chromatid. 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 How many we got?
8 chromatids.
This total one is one.
So 4 is maternal and 4 is paternal.
Here total this one is one chromatid. From here to here it is one chromatid. From here to here it is one chromatid.
Yes.
Okay. So total we got 1, 2, 3, 4. Okay.
Yes, sir.
Total how many chromatids we got?
4.
4 chromatids. Okay. We got 4 chromatids. So in this 4 chromatids, these 2 chromatids, these 2 chromatids are
So 2 chromatid maternal chromosomes and 2 is paternal chromosomes.
these are chromatids of paternal. These are chromatids of
Maternal.
maternal. So maternal chromatids is 2, paternal chromatids is
2.
so total how many we got?
4.
So total we got 4. Okay. How many we got? 4. That's why it is called tetrad. This is called tetrad.
Tetrad.
Tetrad means 4.
4.
Okay. So these 2 chromatids are called sister chromatids. So these 2 are called.
One is maternal and one is paternal. Is called sister.
Both maternal and paternal both these 2 chromatids are called sister chromatids.
Yes.
This one, third one, and fourth one, sister chromatids. First one and second one, sister chromatids. Okay.
Yes, sir.
3, 4 are sister chromatids, 1, 2 are sister chromatids. Okay. Suppose if you take 2 and 3, take 2 and 3, these are called non-sister chromatids. Suppose if you take 2 and 3, they are called non-sister chromatids.
Chromatids.
2 and 3. Like
Sir, but, sir,
one,
but,
yeah, tell me.
sir, but you tell me, first and second is sister chromatid.
Yeah. First and second is sister chromatid. Why? Because
And
both
third and fourth is sister chromatid.
sister
Sir, but,
chromatid. Suppose if you take 2 and 3, or if you take 1 and 3, or if you take 2 and 4, they are
but here 2 and 3 is not sister chromatid. Why?
non-sister means this chromatid is maternal chromatid. This is paternal chromatid. Second one is maternal chromatid. Third one is paternal chromatid.
Yes, sir.
Okay.
Okay, sir.
Suppose
Yes,
if
sir.
you take both maternal chromatids, they are sister. Okay. If
Yes,
you take one paternal and one maternal chromatid, they are
sister chromatid.
okay, non-sister.
Two and
That's
three
why I
not.
mentioned that like this. Suppose even if you take 1 and 3, if you take 1 and 4, they are non-sister. Like if you take 1 and 3, they are non-sister.
Sir, why?
Okay. So 1 is maternal, 3 is paternal.
Yes, sir.
Okay. Like
Sir,
if you take,
maternal and paternal is both is not sister chromatid.
yeah, yeah. Okay. So these all are, if you take these numbers, these all are non-sister.
Yes, sir.
Okay. If you write sister chromatids, only 1 and 2 are sister, 3 and 4 are
Four.
sister.
Sister chromatid.
These are sister. These two are these non-sister. So here always crossing over will take place between non-sister chromatids. Crossing over will occur between
Not sister chromatid.
non-sister chromatids. So in crossing over, so in the process of crossing over, so what will happen in the process of crossing over? So in the process of crossing over, the next step is exchange of chromatids. The next step is exchange
Exchange
of
of chromatids.
chromatids. So this exchange of chromatids will occur between non-sister chromatids. So exchange of chromatids will occur between
Non-sister chromatids.
chromatids. The exchange of chromatids will take place between always between non-sister chromatids. So we'll write the next step. We'll write the next step. So that next step is exchange of
Chromatid.
next step is exchange of chromatids.
Chromatids.
So here what will happen in the exchange of chromatids? What will happen in the exchange of chromatids is okay, I'm drawing again the diagram. Okay,
Okay, sir.
I'm drawing again the same diagram, same like sister and non-sister.
Sir, it will be take place in non-sister chromatids.
Which one?
Sir, exchange of chromatid is take place non-sister chromatids.
Yeah, yeah, it will take place between non-sister chromatids. Okay. So that darkened one are sister chromatids.
Yes, sir.
This left out one are these are sister, these are sister. Okay.
Yes, sir.
So in exchange of chromatids, what will happen is the chromatid, suppose just think that the exchange of chromatid has taken between 3 and 2 and 3
Non-sister chromatids.
chromatids. So here exchange of chromatid has taken place between second and third chromatid. Second and
third chromatid.
third. So what will happen here is the part of second chromatid will break and a part of third chromatid will break. I have drawn a line where they will break.
So second and third chromatid is break.
Will break. Here they will break. After breaking, after breaking of chromatids, after breaking of chromatids, so they will be exchanged. Exchange means this part will go to this. This broken part will go to
Here.
this. Okay. So that means this part will go to this part will go to this means, so what will happen is, it looks like this. So if exchange of chromatids takes place, exchange of chromatids takes place, okay, if exchange of chromatids takes place, okay, so what will happen is, it looks like this. So that means half of the second chromatid will go to third chromatid. So half of the second chromatid will go to
Third chromatid.
third chromatid. Okay. And third chromatid will go to
Second chromatids.
chromatid. Here you are seeing the difference. Here this part has gone to here, this part has gone to here. Okay. So here see, this is totally broken. Okay. Here the darkened part has went here and non-darkened part has come here. You are seeing the difference between these two diagrams.
Yes, sir.
You're able to see the difference between these two diagrams. Here, see this point carefully. Here, exchange of chromatids has taken place.
Sir,
Exchange
but
of
here below side is not the change
here,
chromatids.
this one. See this one, second and third. Don't see other one. Only see second chromatid and third chromatid. Don't see first and fourth.
Sir, second and third below and above
No.
change.
Just one point. See, just here only below point only see. Don't see in the upper point.
Okay, sir.
Okay, just see below point. Okay.
Okay, sir.
Where here this part has gone to here. This part has come to here. So that is called exchange of chromatids. That is called
Exchange of chromatids.
chromatids. After exchange of chromatids, the next step is disjunction. The next is
Disjunction.
disjunction. Okay. Disjunction means the homologous chromosomes start separating. Okay. It is opposite to synapsis. In synapsis, homologous chromosomes has come together, joined, attached with each other.
Sir, how disjunction
Disjunction
homologous
means
chromosomes
homologous
separate.
chromosomes start separate. Yeah, they start separate.
Okay, sir.
So they start separate means so it
Chromosomes just divide.
okay, chromosomes will divide gradually. All of a sudden they will not get separate. All of a sudden they will not get separate. So they will get separate gradually. So now see here they are not joined. Okay, here they are getting separated.
Yes.
Joined. So this separation of homologous chromosomes is called disjunction. Homologous chromosomes start,
Disjunction.
yeah, they separate. That is called disjunction. That is called disjunction. So you understood what is meant by disjunction?
Yes, sir.
Okay. You understood what is meant by exchange of chromatids?
Yes, sir.
Okay. So synapsis also you understood.
Yeah.
You also understood synapsis.
Yes, sir.
Okay. So till now what we discussed is the process of crossing over. So till now what we discussed is the
Process of crossing over.
crossing over. Okay. And when you come to types of crossing over, again there are types of crossing over. Again there are types
Types of crossing over.
So in these types of crossing over, there are single crossing over, double crossing over, multiple crossing over.
The crossing over is three types.
Yeah, three types. Single crossing over, double crossing over, multiple crossing over. Okay. Single,
Yes, sir.
double, multiple. Okay. So single crossing over means crossing over has taken at only one point. Till now what we discussed is single crossover. Okay. So what I told in the process of crossing over is crossing over has taken at only one point. At only one point means only this point crossing over has taken place. Okay.
Yes,
Crossing
sir.
over has taken at only one point. Okay. And double crossing over means if double crossing over means if crossing over has taken at 2 points. Double crossing over means so the crossing over
2
has
points.
taken 2 points. 2 points means this, for example, you can take. Suppose here also crossing over has taken place. Here also crossing over has taken. Okay. Here also 2 points. Okay. Another one that is multiple crossing over. Multiple means if crossing over has taken more than 2 points. Over has taken
More points.
more than 2 points. So it is called multiple crossing over. So it is called
Multiple crossing over.
multiple crossing. So there are three types of crossing over: single crossing over,
Double crossing over, multiple crossing
double
over.
crossing over. Okay. Single means crossing over has taken at 1 point. Double means crossing over has taken at
2
points.
points.
Okay. Multiple means crossing over has taken place at more than 2 points. More than points. Okay.
Yes, sir.
So we discussed about now up to till now it's about process of crossing over and types of crossing over. So now we will discuss about factors influencing crossing over. So what are the factors which will influence crossing over?
Sir, factor influence crossing over means?
That means factors which will promote crossing over, which will increase crossing over.
Okay.
Okay. So the factors which will increase the crossing over is, so the first very important factor distance between the genes. Distance between the genes.
Yes.
Okay. Suppose if the distance between the genes is more, so crossing over will be more. So distance between the genes is more, crossing over also will be more. Okay. Suppose there are two genes A and B.
Sir, here crossing over is over move means crossing over
Yeah,
exchange.
exchange. Okay.
Yes.
Suppose I have mentioned two genes A and B.
Yes, sir.
Okay. Here the distance is like, suppose if A and B are present close. Okay. Here the distance between these two genes is more distance.
Yes,
Here
sir.
less distance.
Less distance.
Okay. So here crossing over chances are more. And in this case, crossing over chances are
Less crossing over.
less. So here if the distance is less, the crossing over is also less. If the distance between genes is more, the crossing over is also
So move.
more. So more distance between the genes, that is distance between the genes is high, crossing over also will be high. If it is less, crossing over also will be
Move.
few. And next, another factor is temperature. Another factor is temperature. Okay. If there are drastic changes in the temperature, drastic changes means temperature becomes very high, very low. Okay. If there are drastic changes in the temperature, it increases crossing over.
Hello.
Okay, drastic changes means, for example, in summer it is
Sir, can you repeat the temperature?
yeah, drastic changes means, suppose in summer it is 50 degrees centigrade. Suppose in winter it is minus 10. There is a lot of difference in the temperature. Okay.
Yes, sir.
This is called drastic change. This is called drastic change.
Take change.
Okay. If this change is drastic change of temperature is there, it will increase crossing over. So it will increase crossing over.
Sir.
Okay. And next factor is X-rays. Okay, X-rays. Okay, X-rays also increase crossing over. X-rays
Crossing
increase
over.
crossing over. Suppose if they are exposed to X-rays, chromosomes and genes are exposed to X-rays means, so the crossing
Increase of crossing over.
will increase. Okay.
Yes, sir.
And another factor, even if they are exposed to mutagens. Mutagens means substances which cause mutations. Substance which cause
Mutation.
mutation. So if they are exposed to these mutagens, crossing over will increase. Crossing over will
Increase.
increase. So these are the factors which will influence the crossing over. The first factor, distance between
Distance between two genes move.
next, drastic change in the
Temperature.
temperature. Next, X-rays.
X-rays.
Next one is
Mutagens.
mutations. So these are the factors which will influence crossing over. Okay.
Yes, sir.
And the last topic, crossing over, is significance of crossing over. It's importance of crossing over. So why crossing over is important? Why crossing over is important? Okay. So crossing over is important in bringing recombinants. Recombinants means new combination of genes.
Yes, sir.
Okay. Yesterday I told linkage and recombinations. So bringing new combination of genes. Bringing new combination of genes. Next, crossing over leads to variations. It leads to variations. Variations means changes in the genes or chromosomes. So these variations in the genes are chromosomes.
Yes. So change of chromosomes.
Yeah, changes in the genes or chromosomes, they're called variations.
Yes, sir.
Okay. And these variations are, so the variations are the important. So variations are required for, so they are important for natural selection. And natural selection means comes under evolution. Natural selection comes under evolution.
Yes, sir.
The natural selection means here the nature will select the fittest one. The nature is a big selector. Okay, nature will select the organisms. Organisms which can survive, they will survive in the, they will get on in the nature. Organisms which cannot survive in the nature, they will die.
Yes, sir.
Okay. And next, crossing over is used to prepare, is used to prepare chromosome maps. Is used to prepare
Chromosomes map.
chromosome maps means, so we can draw chromosomes. There will be a chromosome drawings will be there. And what genes are present on chromosomes. Like this. Okay. Chromosome maps means how many chromosomes are there, what genes are there in each and every chromosome. So that is called chromosome map. Okay. Or even we can call it as
Linkage.
linkage maps. Even we can call it as a linkage map. So this is the importance of uh, the crossing over. This is the importance of
Importance of
crossing
crossing
over.
over.
Okay. So what are the steps in crossing over?
So bringing recombination, variation, important for natural selection. Prepare chromosome map.
No, what are the steps in the process of crossing over?
So process of crossing over.
How many steps are there in that?
So process of crossing over is synapsis, tetrad formation, exchange of chromatic disjunction.
What will happen in synapsis?
Synapsis is the homologous chromosomes attached each other. These are called synapses.
Okay. What will happen in tetrad formation?
So tetrad formation is two chromosomes are combined.
Two chromosomes. Tetrad formation means here four chromatids are formed.
So four chromatids is formed.
Yeah. Tetrad formation means four chromatids are formed. Am I correct? Tetra means how many?
So tetram is four.
Yeah. What will happen in exchange of chromatids?
So exchange of chromatid is will be take place is non-sister chromatid.
So it will take the non-sister chromatid.
Yes, sir.
Exchange of chromatids. It will
So
take
second
in the
and
non.
third so second and third will be break after breaking.
Okay. What will happen in disjunction?
So disjunction is a homologous chromosome start separate.
Okay, they will start separate. Okay,
Yes.
homologous chromosomes will start separate. How many types of crossing over are there?
So three types of crossing over: single crossing over, double crossing over, and multiple crossing over.
Single crossover, double crossover, multiple
Multiple
crossover.
crossing. So single crossing over has taken only the 1 point,
At one point. Okay.
and double
Cross-
crossing over has taken in 2 points.
okay. Multiple crossing over.
Has taken more than 2 points.
Okay. So you have no doubts in this crossing over.
Yes, sir.
Okay. You are clear with this crossing over and telescopic mutations, linkage. You are clear? Any
Yes,
doubt?
sir. No, sir.
No linkage, no doubts.
No, sir.
No mutations, gene mutations, chromosomal mutations. Any doubts?
No, sir. It's clear.
It's clear. Okay.
Yes, sir.
Okay. We will meet you in the next class, tomorrow's class.
Okay, sir. Thank you,
Okay.
sir.
Okay. Bye. Good night.
Good night, sir.
Good night.